Kia ora, I'm David, and thanks to some generous support from the Marion Rhodes Scholarship, I've received the awesome opportunity to perform research in the Cancer Genetics Lab at Otago University. Under Professors Mick Black and Harry Guilford, we're going to be looking at a new technology for sequencing circulating tumour DNA. Circulating tumour DNA is leaked into the blood by cancer cells and has caused a great deal of excitement over the past few years, as it may allow us to learn a great deal about a cancer's genetic and biological state with just a simple blood test. It has been an area of intensive research over the past few years, and companies like Grail are already bringing products into the clinical setting. The problem with ctDNA is that it's incredibly dilute and mixed up with normal DNA, so we need to be able to tell the difference between the two. Genetic mutations in cancer, while common relatively, are still quite rare, so we hope to look at methylation marks, which are much more common through the genome and may allow us to be able to detect ctDNA more sensitively. That's why we are looking at Oxford Nanopore sequencing, which allows sequencing of genetic mutations and methylation in a single run without having to do the old school method of bisulfite conversion. Essentially, my project involves doing bioinformatic analysis of nanopore data and comparing it to other methods to see if it might be viable. If it is, it would offer a number of advantages in the clinical setting. It's quite cheap, and the setup is very small and portable, meaning hopefully we could have sequencing setups in more rural clinics where people could get a blood test and get their cancer state checked up on without having to commute to a large urban hospital. To sign off, I'd like to thank my supervisor for his incredible patience and the people at the OMRF and Marion Rhodes Scholarship for providing me with this awesome opportunity.